Okay, the next half of this lecture is part 2 of the lecture. And what we're going to discuss is cellular reproduction. All right, so cell division is a term that we use to describe exactly what it sounds like, how cells divide, but it also is referred to as cellular reproduction. So cells can reproduce themselves, replicate themselves, or in other words, divide. So cellular reproduction and cellular division are kind of flip-flopped at times, but either way, remember from chapter 1, reproduction is a characteristic of all living things. Okay, therefore reproduction is going to be found on a cellular level. Okay, and that brings in the cell theory and also some other aspects that are necessary for life, to continue life, to maintain a physical body. All right, so if you remember, the cell theory says all living organisms are made of cells, at least one cell, and all cells come from pre-existing cells. Okay, so there's 3 forms of cell division. Binary fission is the first one. Mitosis, meiosis. Binary fission is a method used by prokaryotes. Prokaryotic cells, they reproduce this way. All right, so let's see exactly what's happening here. So this is the parent cell. Okay, I'll go ahead and label that real quick. This is a parent cell, meaning it's the original cell that it's about to be replicated, reproduced, and or divided, same thing. All right, so before this cell can divide, a couple of things need to happen. We can't just cut a cell in half. That would destroy the chromosome count. All right, so what happens is the chromosomes of this organism have to be copied. So see this little coiled up mess? Okay, of course, those are the chromosomes. It's DNA. So every single DNA nucleotide has to be replicated in its entirety. All right, so that's happening here. And when we get to step 2, or step 2, you can clearly see that there's one set of chromosomes right there, and there's the other. And on top of the duplication of chromosomes, the actual cell itself has to grow. So it's growing in length. See how long it is compared to the original parent cell? Right. All right, so it'll continue to elongate, and then it'll start to split. See how it's making this kind of inversion? It's starting to divide, and this will keep happening, and it'll get closer and closer until we have split the cell in half, and we have 2 genetically identical daughter cells. All right, now that terminology is really important. 2 genetically identical daughter cells. All right, very good. Okay, so what about eukaryotes? That's how prokaryotes reproduce. What about eukaryotes? Well, this is where mitosis and meiosis comes in. So mitosis is one type of division, meiosis is the other. Now, what's the difference? There's a massive difference. So here we go. So mitosis is designed, or what it actually does is to take our cells, or cells in general that are eukaryotic, and reproduce them. Now, why would we need that? Why would we need to generate new cells? Well, we do. We need the generation of cells for growth and development. Okay, and we need it for any kind of cell renewal and cell repair. So you have cells that divide every 24 hours, your skin cells. You have cells that get damaged and they need to be repaired. That's through mitosis. Okay, now look at this term. This is important. It says mitosis generates new diploid cells. Remember diploid? Diploid, D-I, di. Okay, diploid means that the cell has the full set of chromosomes for that species. Okay. All right. So let's do an example. Humans, for example, our full set is 46 chromosomes. Okay. And when we write it out, it would be, that means diploid. Okay, so, alrighty, meiosis. Okay, meiosis is used for one reason and one reason only, for the production of gametes. Gametes are the sex cells. Okay, so see, I want you to know this term, gametes, because I'm going to use it. But these are the sex cells, like eggs and sperm. The reproductive cells. And notice that it says meiosis generates new haploid cells. What does haploid mean? Okay, haploid means that the organism has half the number of chromosomes for that species. Example, let's say we're talking about a haploid sex cell. Okay, so I'm not going to really write anything other than this. So the count, the chromosome count should be 23. Okay, so in other words, eggs and sperm, each one is carrying 23 chromosomes. All right. So what we're looking at here, this is called the human karyotype. So scientists have figured out what our chromosomes look like. They basically figured out all of our genes, and through the Human Genome Project, they know what they do, what they encode for, and things like that. Now, what this is your human karyotype. So even though each person has unique DNA, it doesn't really matter that our DNA, well, it matters, but in regards to the human karyotype, your chromosomes look like this as well, even with your unique DNA. So what we're looking at here is a layout, a display, a physical display of what our chromosomes look like. Okay, notice that each one has a number next to it. So there's number 1 all the way to 22. And then these are called autosomes. Autosomes. Okay, now the 23rd chromosome, of course, is the 23rd set is the sex chromosomes. All right, so earlier I said that humans have 46 chromosomes, and we do. And this is what they're supposed to look like. So you really have 2 sets, or 1 set. So 2 chromosomes at each location. There's chromosome 1. That's another chromosome next to it. Okay, so you have these are called homologous chromosomes. So you have homologous chromosome set number 1 right there. Okay, that's another chromosome. That's another. Okay, so you have 23. If you go through all of these, 23 pairs. And then, of course, if you are female, you're going to have the XX chromosomes. Males have the XY, and that will determine the sex of the person, the baby, the individual. Okay, now, if you count them all up, 23 sets, that's 46. All right, now, another important factor here, and this should be something that you basically take home and remember permanently. You inherit 23 chromosomes from mom and 23 from dad. So one of these, we'll just say the first one, okay, is the mom chromosome. Okay. The second one is the dad chromosome. This one's maternal, paternal, maternal, paternal. So either way, you get a total of, when you inherit your chromosomes, when you're born, when you're made, right, you get 23 chromosomes from mom, and you get 23 chromosomes from your dad, giving us a total of 46 total chromosomes, right? And then also I want you to see this term as well. And really what it is we have 23 pairs of, okay, make sure you're taking notes. This is homologous chromosomes. All right. Very good. Let's go ahead and move on. Now, inside of a cell, I'll come back to this picture. Now, let me just kind of recap what we just discussed. Now, this is the human life cycle. Okay, here we go. Okay, now, every single individual starts off life this way. Okay, you get 23 chromosomes from the mother. Okay, notice this says haploid gametes. That means that there's 23 chromosomes in this egg. Okay, that's the mother. So this is technically n equals 23. So they expect you to just know that it's haploid when they use this, when you see n. Okay, so there's 23 chromosomes in that egg. There's 23 in that sperm. Okay, they're going to fuse together through fertilization. And everyone begins life as a single celled diploid zygote with 46 chromosomes. So yes, it's 2n equals 46. All right, so how do you go from being this little tiny single-celled zygote to a full human adult made of trillions of cells? Through mitosis. Okay, so we get from here to here, in the majority of the part being mitosis. Of course, there's other factors involved in growth and development, but mitosis is key. Now, every single one of your cells, every single one except red blood cells, is carrying your full DNA that you inherited or that you acquired in this process right here. So this DNA is, okay, let's say this is you. Okay, that's your genetic makeup right there. That DNA is found in every single cell because of mitosis. Because mitosis takes cells and copies everything before it moves on. Okay, now we're going to learn how that happens, but this is important to kind of put together big picture stuff when it comes to cell division. So mitosis is used for growth, development. Okay, and then once you're an adult, you need cell renewal. You need cell repair sometimes. Alrighty, so mitosis is responsible for that. Now, meiosis is for one reason and one reason only. Okay, in the female body, this happens in the ovaries, and the only purpose of meiosis is to make haploid gametes. That's all. That's the only reason for it. Now, it's a big process, but I want you to make this distinction now. Okay, in meiosis in the male body, this is the production of the haploid gamete sex cells occurs in the testes, of course, and then that is the purpose of meiosis. Meiosis has one purpose and one purpose only: to generate the sex cells, to generate haploid, okay, generate haploid. What's wrong with me? Haploid. There we go. Generate haploid gametes. That's all. All right. Now, let's go ahead and look into a couple of important factors before we jump into the steps of mitosis. Basically, let's talk a little bit about DNA. Now, in chapter 4, I mentioned that DNA, when it's unraveled, is called chromatin, or referred to as chromatin. Now, I want you to understand the structures of chromatin and what chromosomes are, and this is a new term called sister chromatids. All right, so this shows quite a bit. All right, so here's the DNA. It's the double helix, and notice that it's uncoiled, okay? And it's wrapped around these proteins called histone proteins. So this is chromatin. All of this uncoiled stuff. There we go. Okay, now chromosomes coil up and they make that chromosome shape that we saw earlier, like this. Okay, look at them one at a time. Okay, so see this here? This is one chromosome. Okay, I'm drawing around it. That's one chromosome. All right, now when cell division is happening, it can't just happen. Remember, with the binary fission, what was the first thing that needed to happen? We had to copy all of the prokaryotic DNA. Okay, well, eukaryotes work the same way. They're going to have to copy every single chromosome that we have, and in order to reproduce a replicate cell. All right, so once it's done so, okay, once it's copied itself and basically made a complete duplicate of itself, that is called a sister chromosome, or I'm sorry, sister chromatid. It's a replicate chromosome. Okay, so in other words, every chromosome that you have is going to be copied. Okay, it's duplicated, and on top of it being duplicated, it's also held together with a protein. That protein is called the centromere. Okay, and now this is an exact replica of this, and they're held together, and now they're referred to as sister chromatids. You want to know what that term is because you're going to see it quite a bit. Okay, so for a time, the sister chromatids will be held together by a centromere, and then at some point they will just be distributed to daughter cells, to two daughter cells. All right, So that's that. Now I think we're ready to talk about the cell cycle. Okay, so the cell cycle is a cycle that every single cell has to go through in order to divide or replicate. So basically, I want you to understand this very well and pay close attention. All right, so look at this pie chart here. See all of this area? This whole thing goes all the way around. It takes up most of the cell cycle, and it takes up most of this chart. Okay, because it is the longest part of the cell cycle. It's called interphase. Okay, now the second part of the cell cycle is called the mitotic phase. See this little blue slice of the pie? That is the mitotic phase. Okay, so let's start with interphase. So if a cell is going to enter the cell cycle, let's say a cell is going to divide, it has to be instructed to do so. Cells cannot just go into the cycle and start dividing without any control, because that is the exact definition of cancer. Cancer is uncontrolled cell division. So normal, happy, healthy cells that don't have a problem, that aren't cancerous, they follow these steps. Okay, so basically, the first thing is a cell has to be instructed to enter the cell cycle with growth hormone. Okay, growth hormone does exactly what it sounds like. It is responsible for growth. Okay, and that includes cells and their replication process, right? All right, so growth hormone is released and the cell will enter the cell cycle, and it will enter the first part of the cell cycle, which is called the G1 phase. Okay, what happens during this phase? Well, the cell's going to have to grow. Okay, during the G1 phase, what's going on is that the cell is growing and in size. So the plasma membrane is growing, let's see, I'm going to add some things to this list here. Let's move this down. There we go. Okay, so G1, what's happening here? Aside from growth, this is a primary growth phase. Now, the things that are going to grow are everything, okay, except for DNA. The plasma membrane is going to grow. The cytoplasm will grow, okay, and all of the organelles, all of them, are duplicated. Okay, everything. I mean, the rough ER, the smooth ER, all of the mitochondria, the Golgi, all of these guys have to be copied, because at the end of the cell cycle, we're going to get two identical cells. How do you get two identical cells? Well, you have to copy everything, and I mean everything. All right, so all of the cytoplasm, the cell membrane, all the organelles, proteins, etc. are going to be duplicated, okay, in G1. Now, once that cell has left the G1 phase, it will move into the next phase. Okay, the next phase is called the S phase. S stands for DNA synthesis. Okay, so everything is duplicated and copied here. Now in G1, now in the S phase, the only thing that's copied is DNA. Okay, and once DNA is copied, everything will be duplicated. All the DNA will be duplicated. So let's say we move into the next step, the G2. How much DNA do we have if this were a human cell? Okay, well, if this was a human cell, a normal human cell, right, then it should have how many chromosomes? 46. Should have 46 chromosomes. Okay, now as it enters the S phase, it will have 46 chromosomes. Okay, when it's done with the S phase, where all the DNA has been copied, every single chromosome, and it's going to be duplicated, and it's also going to be connected to its sister chromatid. Every single chromosome is copied and clipped together with a protein, and they're sitting now in sets of sister chromatids. And actually, it's more like this. All right, so this is a human karyotype after the S phase. Everything has been copied. Okay, so notice that there's no longer 2 chromosomes at each location. There's 1, 2, 3, 4. That is that. Everything has been duplicated. Notice that these 2 are connected. They're sister chromatids. That means these are sister chromatids as well. So are these, so are these, so are these, so are these. And it goes all of them. Every chromosome is copied and clipped together with its duplicate. All right, so that's a big process. That's the S phase. Now, the G2 phase is when a cell enters G2, it's going to have double the number of chromosomes. So it's going to have 92 chromosomes here. And this is the final growth phase before cell division occurs. And cell division, the G2 phase is going to check. There's going to be a check point in there where the cell is going to be checked for errors, making sure that there are no errors, that every single chromosome was copied, that every single thing occurred properly, all the organelles are duplicated, it'll grow a little bit more, and then ultimately what's going to happen here is that the cell will enter the next part, which is the mitotic phase. The mitotic phase is broken into 2 parts. Mitosis, right here, and cytokinesis. They all make up the mitotic phase. Okay, now here's the deal. Do you see how small the gap or the part of this cycle mitosis is? It's tiny. It's this little tiny sliver in this big cycle. So I want you to know the cycle completely, every single part of it, and what is happening in each phase. Now let's look a little closer at the mitotic phase. Okay, the mitotic phase is 2 parts. Now actually, really quick, let me just make this even more clear. Interphase, if I asked you, how many phases does the cell cycle have? What you would tell me is it has 2 parts. It has interphase, number 1, and the mitotic phase is number 2. That is in reference to the cell cycle. Now, if you were asked, what are the phases or subphases of interphase, then you would list off G1, S, G2, and then of course you have to know what's happening in each phase. Okay, now the mitotic phase is broken into 2 parts, mitosis and cytokinesis. Okay, so it's 2 parts. Now, mitosis, what that actually means, the definition of mitosis is the nuclear division, meaning division of the nucleus. We're going to divide up the duplicated DNA and distribute them to 2 daughter nuclei. A nuclei is capable of dividing in half. So those terms right there are incredibly important. All right. mitosis is nuclear division. Cytokinesis is when the cytoplasm divides and finally forms two daughter cells. Two genetically identical daughter cells. All right, but we'll get into the details of that. Now, mitosis is this subphase of the mitotic phase, also referred to as the M phase. So sometimes the book will just call it, they'll call the mitotic phase the M, okay, the M phase for short. So if you see that, it means the same thing. All right, now mitosis is part of the mitotic phase, and the mitosis has several steps. It has prophase, something called metaphase, anaphase, and telophase. Okay. Now I want you to, we're going to learn each one, but quickly, I want to emphasize something. A good way to remember these steps is like this. PMAT. PMAT. Prophase, metaphase, anaphase, telophase, PMAT. Okay, and that's a pretty easy way to remember it. Okay, this is the first part of mitosis. So the book will sometimes tell you that there's 5 phases, but we have condensed them into 4. The video will tell you that too. So basically what they're going to say is that there's a prophase and that there's a prometaphase. So what we've done is we've taken prophase and prometaphase and put them into one category and just called it prophase. So all of this is happening in prophase. All right, so what we're looking at here is clearly a diagram cell. And then this is a real cell, and it's been fluorescently stained. Just show you this real quick. And it's fluorescently stained so that we could see everything. Okay, so this is the cell, and all of this blue stuff right here, these are the chromosomes, okay? And this is where the spindle is going to form on each side. All right, so if we look at the parallels of the picture along with the diagram, let's take a look at what's going on here. Okay, so there's certain things and ways to remember all of the steps. And this is how. In prophase, the following occurs. Something called the mitotic spindle is going to form. What is that? Well, that's this. See these little guys right here? These are called the centrioles. Okay, and they're not usually in that position. See them in green up here? Yeah, okay, they're not usually in that position. During prophase, they get into that position, and not only do they do that, they make this net. See this kind of, it almost looks like a net, okay? This is called the mitotic spindle, okay? And both sides are going to make it because we have 2 poles. Look at the cell like an equator. So when we say pole, we mean north and south, okay? So we have the mitotic spindle formed. The centrioles, which are these, are going to migrate to the poles of the cell. All of our chromatin is going to condense into visible chromosomes. So normally we cannot see DNA, okay, unless a cell is in the middle of cell division. Then you can see it. Clearly you can see it, and you can see it right here in this image. Okay, right there, all that DNA, you normally wouldn't be able to see it. Okay, but we're in prophase, so you can. Now, the other thing that's happening is we have a nuclear membrane that surrounds our DNA. Remember, our DNA is not floating around like a bacteria cell. Instead, no, it's encapsulated in a nucleus. So how does a mitotic spindle get its little hands on these chromosomes? Is because the nuclear membrane is fragmenting. It's disappearing. It's disintegrating. So that's something that happens in prophase. Okay, so the nuclear membrane is going to dissolve. And generally the cell will kind of slow down on any metabolic activity. Metaphase. During metaphase, now I want you to pay attention to this. This is pretty interesting. Between prophase and metaphase, we've come a long way. Now, what's happening here is the centriole and its mitotic spindly arms are connected to one of the sister chromatids on this side, right? And this one is connected to these sister chromatids on this side, okay? And that goes for all the chromosomes. Now, there's that. And they have full control over the chromosomes right now. These are technically sister chromatids. And what they're going to do is push and pull and move and align all of the sister chromatids at the equator of the cell. That's where they are. Now, if we look at this picture, which is actually a great picture, you could see it. See all the chromosomes? Right there, aligned, and yeah, they kind of look like they're way up here, and because it's DNA, but they are aligned at the chromosome. At the equator. And then this is one centriole. You can clearly see the mitotic spindle in green. Centriole, mitotic spindle. Okay, very good. So metaphase is known for one distinction. All of the sister chromatids line up at the center of the cell or the equator of the cell. Okay, anaphase. All right, so what happens here? Pretty straightforward. This mitotic spindle was not always there, right? So it grew out of the centrioles, and in turn, it can retract or shorten and move this way. Okay, now if both sides, the North Pole centriole and the South Pole centriole, their mitotic spindle shortens on both ends, it's going to pull the sister chromatids apart. And that's what's happening in anaphase. And what's cool is you can see that happening right here. See how they're pulled apart? And look at this, the mitotic spindle shortened. Perfect. All right, so anaphase is when sister chromatids are pulled apart and or separated. And then on top of that, now they're going to move towards the poles of the cell. So they're going to move to opposite ends of the cell. One will move to this side, okay? And one set will all move to this side. So I guess we can grab all of these. They're all going to go this way. All right, so next, telophase. Well, now the chromosomes have been pulled apart and they've been pulled to opposite ends of the cell, and so we don't need the mitotic spindle anymore. It can disintegrate, and so it does. And then our chromosomes don't need to be in the form of chromosomes. They can uncoil and unravel and go back to chromatin, and so they do. Okay, and then we're going to need our nucleus back. We can't have DNA without nucleus for long. We can only allow it, or the cell will allow it during, the cell division process, but it's coming right back, and there it is. You can see these little fragments. The nuclear membrane is reforming around the DNA, okay, on both sides. All right. Okay, and the cell is starting to divide. See this? This is called the cleavage furrow. This kind of this whole the cell's kind of folding in on itself, and really it's not folding. What's happening is we have these little contractile rings, and they're going to keep contracting, and they're going to keep pushing and pushing and contracting until the cell, this is going to keep moving this way, and this one will keep moving this way until they cut the cell in half. But that doesn't happen yet. We're still in telophase. All right, so that's the end of mitosis. Okay, remember PMAT, prophase, metaphase, anaphase, telophase. Now we're going to look at cytokinesis. Now cytokinesis is when it technically is going to happen simultaneously with telophase because it happens so fast, but the fact is that cleavage furrow is going to form, and those contractile rings are going to cut this cell in half. And we're going to end mitosis with 2 daughter cells, and one of the most important parts is there are 2 genetically identical daughter cells. That means the DNA here and here are exactly the same. And that is the reason why every cell in your body has the same DNA, is because of the cell cycle and because mitosis is so precise, and it typically doesn't make mistakes. Mistakes can be made though. Okay, now plants are eukaryotic, if you remember. Therefore, their cell cycle is not different than our cell cycle, right? The only difference is the last part, the cytokinesis. So when our animal cells divide, they're going to have a cleavage furrow. When plant cells divide, they're going to have a plant cell wall forming, and really it's called a cell plate for now. Okay, so basically what happens is, don't forget this is all made of cellulose, this plant cell. Let's take a look. All right, so this is all made of cellulose. It's a cell wall. So we need these vesicles to drop off this material to make the cell wall. Okay, so we have these little fragments of the cell wall, and they'll fuse into a plate temporarily until they keep growing. Okay, they'll grow this way and this way until we have a completely new cell wall and 2 genetically identical daughter cells. And that is mitosis. Okay. So, you want to know everything about interphase.
During the mitotic phase, a cell will undergo mitosis to form 2 new nuclei, and then divide to form 2 new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into 2 new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and two centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands, called microtubules, appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear
All right. Now, can any cell enter the cell cycle at will? What are the rules and regulations to cell development and cell growth? Okay, so there's a couple. Let's start with this one over here, okay, to the left. Okay, this is called Anchorage Dependence, and there's also 3 other factors: a cell density factor and a chemical growth factor. Okay, and we've already talked a little bit about growth factor. Okay, so it's actually, yeah, here we go. So let's actually look at both sides to begin. If you take cells, okay, and you suspend them, you put them in liquid. So see all of these cells, they've been placed in this cultural cell suspension. And basically what that means is it's got all of the nutrients and factors that are required for a cell to survive. So they have all the food and nutrients that they need, right? So they can divide if they need to, but they're floating around, meaning that they're not anchored to anything. So they don't actually grow. So if we count up the cells, okay, so they've been separated here. Okay, there's no treatment. This is a control. And this is an experimental group. Alrighty, so there's that. Alright, so they put 4 cells in here. And if we count them up, there's 1, 2, 3, 4. They waited a certain amount of time, and there's only 4. Okay. Well, that's because cells cannot grow in a liquid. They have to be anchored. Okay, so they took 4 cells and placed them in a petri dish. This is technically, some kind of plastic, but there's an anchorage. They can hang on to something, and so they do, and they will grow. They will grow a perfect layer, and it's a single layer because they know when to stop. When there's enough of cells in that location, they will stop. That's in reference to the cell density control. So that's that. All right. Now let's look at some factors here. Okay. So anchorage is important. Now, the other things that are important. If you remove a cell, density is important. If you remove a cell or 2, they will be restored immediately. And the exact amount of cells removed, so they took out 1, 2. 2 will be replaced because the cells know that there's something missing. They can tell, they can feel one another. All right, now the other experiment. Now we started off taking these cells, right, and putting them in this liquid and in a control with nothing. They won't grow. They're not supposed to because they follow that whole Anchorage Dependence law. Okay, now, but what can happen is this. If we add something called growth factor is a hormone, and we add it to the experiment, to the cells, they will grow. They will grow because growth factor initiates cell division. So if you look at these 2, sorry about that, there we go. Let's look at these 2. We start off with 4 cells, and then we have, a significantly higher number. I don't want to count them all. There's something like, I don't know, we can 10, 12, 15, something like that. All right, so either way, these are the things that affect cell division, meaning that they follow laws, And there's certain things that can break the laws, like the growth factor can break this law, okay, but mostly they have to be anchored to something. And in the human body, they're going to be anchored to your underlying tissue, like they're going to be anchored to your cartilage. They're anchored to something, okay, they're not floating around. And then the cell density is a factor. This whole perfect single layer is an important factor, and the fact that it's restorable is significant. So they don't grow out of control. They know when to stop. And then growth factor, again, has a direct effect on growth. Alrighty, so let's check out this, this whole thing. So we have something called checkpoints. Now, what we're looking at is the cell cycle, but we're looking at something called the cell cycle control system. The cell cycle control system. Okay, what does that mean? That means there's a system that controls the cell cycle. That means that cells cannot just enter the cell cycle whenever they want to. And they can't do whatever they want to. They have to be controlled and regulated, or we would just have pandemonium. All right, so this is how it works. First, let me get an arrow. Hang on, guys. There we go. All right, growth factor. This is a hormone. It has to bind to a receptor protein, and it does. That's the signal. This is the kind of go to the cell cycle signal. It says, okay, you're allowed to grow. Go ahead and enter the cell cycle. So it does. And it enters at G1. And now, see these little, they look like lights, like traffic lights. Okay, there's a reason for that. Okay. This is where the checkpoints are. Okay, so we have a G1 checkpoint. A G1 checkpoint is going to check that the cell is supposed to really divide. So it makes sure that it does. And now, if it gives it the green light and it allows it to move through, it will usually move through the entire cycle, unless there's a problem. Okay, so there's a second checkpoint at G2, the G2 checkpoint. This checkpoint is going to make sure that everything was perfect in duplication. And that means DNA, all of the organelles, it's going to make sure that the cell is big enough. It's going to make sure that the cell is ready for division. And that is the G2 checkpoint. Okay, let's say that there was a mistake. Let's say that the G2 checkpoint caught a miscalculation of chromosomes. There's a there's an error in the DNA synthesis. Okay, the cell has to be destroyed, and it will be. So it's removed out of the cycle. Okay. And it goes into a programmed cell death state called apoptosis. Okay, so this is programmed cell death. It's like cellular suicide, because it could be, let's say there was a mistake in the gene replication, and it could be a mistake that could cause cancer. So it has to be destroyed. So our cells and our body and our system is so sophisticated and savvy that it just keeps track of everything, and it doesn't let anything through the cracks in a healthy system, right? Of course, there's things that can damage that, but that's a different issue. All right, so let's say that the cell, that's apoptosis. Let's say that the cell did well, everything went perfect, and it goes on into the M phase. There's a checkpoint here in the M phase to make sure that during Anaphase, those sister chromatids were indeed divided. Because if they're not indeed divided, we're going to have a big problem on our hands, a huge problem. That's a literal genetic disorder. And then let's say it passes, then it's done. And they'll leave the cycle and go do what it's supposed to do. Of course, if it fails the test, apoptosis it is. Okay, so those are the 3 major checkpoints. What about this one? G0. Now, G0 or G0, this is a non-dividing state. So lots of our cells just sit in this non-dividing state until instructed to do otherwise, meaning they're given some growth factor by the body and told, go ahead and, go to the cell cycle. Now, there's some cells in our body that stay in G0, which is a non-dividing state. Okay. So liver cells stay in there. They can be called back to the cycle by growth factors and things like that when there's injury, but they really don't replicate often. Liver cells don't. And that's why people that drink too much alcohol, like alcoholics, can get cirrhosis of the liver pretty quickly. And it's because our liver cells do not go through the cell cycle very often. All right. Now, yes, the cirrhosis can lead to some liver cells dividing, but it's not going to repair all of them. Okay, now some specialized cells never divide, meaning after they're formed, they never will divide again. Nerve cells, muscle cells, they never divide. That means if you damage a nerve cell, you're done. It will never fix itself. That is why people get paralyzed or nerve damage is permanent, because of the fact that they do not go through mitosis. They stay in a G0 stage. Same thing with the muscle cells as well. They do not leave the G0 phase. The liver cells can, but not very often. Okay, I'm going to actually skip over this. All right, so now we just talked about mitosis, the cell cycle, and controlled cell division. Controlled cell division is very important, but uncontrolled cell division is not. Now, if there's cells that slip through the cracks, which there are, that can lead to cancer. So cells can have damaged DNA. Cells there's a range of genes that can be affected and damaged that play a role in the cell cycle and lead to uncontrolled cell division. So controlled cell division is great. We need it. Okay, but it'll stop eventually when it's done doing its job. A cell that has damage to its DNA, okay, is going to just keep dividing and dividing and dividing and dividing and dividing over and over and over and over and over again. And it won't stop. And then we end up with a tumor. Now, here's something I want you to take away from this whole thing that's significant. Everyone has unique DNA on the planet. Everyone, except for identical twins. That's a different situation, but for the most part, everyone, unless you're an identical twin, you have unique DNA. Okay, therefore, let's say a person gets cancer. See all of these cells, these cancer cells? What do they have in their nucleus? They have unique DNA, every single cell. Therefore, here's the problem. Now, let's say we have a group of people that have breast cancer. Every single breast cancer patient has unique DNA, and their cancer is technically unique. So there are, you know, breast cancer treatments, and there's cancer treatment medications and chemo and radiation, and there's ways to destroy the cells. But what's happening is this is not a virus or a bacteria. These are the host or that person's cells with their DNA. And the problem is finding them, targeting them, and destroying them while not hurting the other cells. Okay, and we really haven't come up with the best solutions yet. I mean, there's chemo, and that hurts everything. Well, not everything. It hurts rapidly dividing cells. Chemo is going to attack all of your cells that are dividing rapidly. Let's say, you know, we're talking about a cancer patient where the chemo will destroy the rapidly dividing cancer cells, but then it'll destroy the rapidly dividing normal cells too. So what are some rapidly dividing normal cells? Okay, your skin, your entire GI tract. That's why chemotherapy patients get sick and vomit. Your hair is made of proteins, and that is rapidly growing, and that's why people's hair falls out and things like that. All right, so if we had a universal, that's why we don't have a universal solution for cancer. We don't have one, because we would have to design a medication for every single human being that has the disease, and nobody has that kind of money. I mean, we're talking hundreds of millions of dollars to even get a specialized type of DNA. A specialized type of cancer medication for one individual. It's just not even possible right now. Maybe someday, I don't know. I mean, they don't put enough money into research, so that's another problem. All right, but if you've ever wondered, why can't we just get rid of this horrible disease? Well, that's why. That's one of the bumps in treating cancer. All right, now speaking of cancer, there's different types. Of course, like we all know this, there's benign and malignant. Ultimately, something that's benign is going to just not invade other tissue. It'll stay like this as a tumor. Okay, now if it starts to invade tissue, like these glands here, that's a problem. Okay, when it has fully metastasized, okay, it gets into your blood vessels and the lymphatic system. Okay, your blood vessels is your network that travels your entire body. So once it's in the blood, it can go anywhere. Now, there are patterns. They've found that it'll usually move to the next organ, but not always. It can get in there, and basically that cardiovascular system is going to be pumping those cancerous cells all over the body. All right, so that's enough of that. Cancer sufficiently. Okay, now this is the end of chapter 3. Be sure to review your study guide and study directly from our video lectures. See you guys next time.